All right, so okay. Now xylem vessels, these are basically elongated hollow tubes that are made of xylem cells linked end to end. Okay. Now xylem cells are dead at maturity. So xylem vessels. Now the ones that I'm writing in blue are the functions, okay? So what are the adaptations to these functions? I think we also talked about this in the first chapter. The red ones are the adaptations. So the two adaptations are basically the absence of protoplasm and cross walls, and another one is the deposition of lignin on the cell walls. What about phloem? The phloem are actually living cells, okay? Degenerated protoplasm means that they lack organelles such as the nucleus, yeah, ribosomes, large central vacuole, but they're living. And there are walls, okay? So with porous walls called sieve plates between them. Now these ones, xylem did not have any cross walls, but sieve tube elements, they do. I mean, phloem. Now about the companion cell. There is one companion cell with each sieve tube element. These companion cells, they do have nucleus, cytoplasm, numerous mitochondria, and are basically, they have everything, okay, that a normal cell has. And they are responsible for performing all the metabolic functions for the sieve tube elements, because the sieve tube elements, they do not have anything. Like I said, they have degenerated protoplasm. So this companion cell does everything for them.
I'm done.
Okay. Now, yeah, I'm going to still write about the phloem, but I was done with the xylem in the previous page, so I'm just going to continue with the phloem. Now, the blue ones are the functions. So what are the functions of phloem vessels? To conduct sugars and amino acids. Okay, sorry. Amino acids from the leaves to other parts of the plant. Now, what are the adaptations to this function? There are porous sieve plates in between, like I mentioned. So these porous sieve plates, they allow uninterrupted flow of food substances through the sieve tubes. That's why they're important. What else? Yeah, the numerous mitochondria in the companion cell provide energy The companion cells are there not just to just to perform all the metabolic functions of the sieve tube elements. So that includes providing them with energy and also helping them you know uh, helping them pass the sugar. So they help them load the sugar into the sieve tube element. Or almost like বাসের হেল্পারের মতো, প্যাসেঞ্জার উঠাইতে হেল্প করে and সবকিছু. So if I draw a diagram of phloem vessels, so so wait, I'm going to explain. I hope you get a picture. Now, obviously, these are the different companion cells, okay? And this plate right here, this is a sieve plate. And each of these, so between two sieve plate and accompanied by a companion cell, this entire thing right here, for example, is a sieve tube element. All right. Yeah, that's it. As you can see, the sieve tube is very porous. Okay, it allows the sugar to pass through, and also these phloem cells right here. Quite sorry, the sieve tube element. It's quite thin, and yeah, it has a degenerated protoplasm. That's it. Done. Are there any questions?
No, okay. I'm done copying this.
Okay. Now, both of these together, so phloem vessels and xylem vessels, both of these together form the vascular tissue, like I mentioned at the beginning. Now, where are they positioned in a dicotyledonous stem? That is what we're going to learn. So the entire thing, the entire vascular tissue, how does it look like? Where is it situated? Let's talk about that. Okay, now in dicotyledonous stems, the vascular tissue or the vascular bundle, they are arranged in a circular ring, okay? Arranged in a ring around the circular pith. Actually, let's draw it first. Remember, this is the stem that we're talking about. Later, we will talk about the roots. So don't ever confuse it. Now, most of the time you might be asked to draw this or even label this. So these are quite important. Even the microscopic pictures of these diagrams, you should be able to identify. Okay, I'm going to show you a few after we're done with this. This is called the pith. The the part that is closer to the boundary is called the cortex. This is obviously the epidermis. And these drop-shaped things, these are the vascular bundles. So this entire thing is a vascular bundle. Okay. Now, what does it consist of? Yeah, so this one right here, let me do it in a different color. Xylem. This one right here is the phloem. And the middle thing right there, this is the cambium. Okay. All of these together is the vascular bundle.
Apu, what's the cambium?
These are, a couple of cells that lie between the phloem and the xylem vessels. So like if I go back to the picture where I drew the leaves, this one. So like somewhere in between there are cambium, there's cambium, yeah.
So Apu, cambium separates the xylem from the phloem.
Yeah, cambium tissue, it is like a layer of tissue.
Yeah.
It is usually found between xylem and phloem. I don't know if their function is specifically to divide them, but yeah, they're usually found between xylem and phloem.
Yeah.
Okay, yeah, for your information, I actually Googled to see what is the role of cambium tissue. It has nothing to do with transport. It actually is a tissue layer that helps in plant growth. Okay. It is like an undifferentiated mass of cells. So later, maybe, I don't know if you have it in O levels, but in A levels, you'll learn about stem cells. So in plant cells, cambium are a type of stem cells, which are undifferentiated cells. Okay, that's it.
Apu, the pith and the cortex, they're like hollow, right? There's nothing there.
In stem, yeah, they're a type of tissue, so there should be, like cells all over, but I didn't draw them.
Yeah.
I'm going to show you a different type. Sorry.
I said, Apu, I was done drawing.
Oh, okay. Yeah. I'm going to draw a different type of diagram, which can come up, and just so you don't get confused. So like if it is drawn like this is the same thing. Yeah, sometimes it can be drawn like this. Then you know clearly that this middle part is the pith. This outside is the cortex. And then this inner thing is the xylem. This outer part is the phloem. And this middle line is the cambium. So this one is the cambium. Yeah, that's it. And the outer part is obviously the epidermis. Just so you don't get confused, it can also be drawn like this, okay? Yeah, you can copy this if you want.
Yeah, I'm done. Yeah, let's show you this picture. I think you can see the lower picture that looks more like from a light microscope. So you will never be able to see anything clearly differentiated. You just have to guess, okay? So the middle, all these cells, all these circles, that's the pith. And then you can see there are, I know the resolution is very bad, but yeah, I hope you can still see. Then there are drop shaped parts. So the inner part will be the xylem, and then out part is the phloem, and then we have the cambium in between, and outside, just before the epidermis, we have the cortex. Okay. Yeah.
Okay, let's write the functions. So yeah, because they're undifferentiated, they can differentiate into anything. They can form new xylem vessels or phloem vessels. It really matters what the plant needs. Okay. food is stored in the cortex and pith. That's it. I'm coming back in a minute. Okay, I'll take 5 minutes because I'm going to pray. Okay.
Yeah, sure. Can I go pray too?
Yeah, sure.